All this is Dr. Mobeen Sayed from Drbeen.com. Welcome to one more show. So we have been talking about IVIg, for example, in long haul patients, and there have been some KoolBeens who have been asking about the IVIg and how does it work. So I thought today, as I've gotten my shot and I would like to make it brief, so I thought today we would talk more about IVIg. And because I have had my shot, I am pretty tired and fatigued as well. So not a lot of, actually no beautiful cartoons, just I'm gonna just doodle while we discuss it, and I hope that that would be okay. uh, so first of all, this is Drbeen.com, and here we are going to talk about the IVIg. I will then do a chit chat as well. I think that actually helps me continue with my time as well, and we would do some more discussions about how am I feeling, what are the symptoms, and something very interesting about the ivermectin, and also some comments on the video. So let's start. Hope everybody is doing fine. Let's start and let's talk about IVIg. So the things that I want to establish today with this talk is, number one, what is IVIg? Number two, how is it made? How is general high level? There are such deep and elaborate processes to make it safe and to have various variations of the final infusion. So what is IVIg? How is it made? And what is the use of it? And what is generally, what is the difference between subcutaneous or subcutaneous IVIg, or not IVIg, but subcutaneous immunoglobulin and IVIg? So that's what we're going to talk about. Let's start. It is going to be a little less structured because I have not already organized it. So please bear with me, and I think we'll be okay. So first of all, IVIg is basically immunoglobulin G. Immunoglobulin G, as we know that when, I'm going to go over this for a second. So imagine there is some antigen. Imagine nowadays we're talking about SARS-CoV-2 all the time. So imagine SARS-CoV-2 enters our body. It enters a cell, let's say a dendritic cell or a macrophage. We know this diagram, all of us, so I don't want to do too much here. But once that antigen is produced inside the cell, then the antigen is shredded. It is broken up into pieces. Those pieces are shown up, or as we call it, they're loaded on MHC1 or MHC2. MHC1 stands for Major Histocompatibility Complex 1. MHC2 stands for Major Histocompatibility Complex 2. MHC1 is a protein that can be present on all nucleated cells, and MHC2 is a protein that is only present on the professional antigen presenting cells, which are three: dendritic cells, macrophages, and B cells. Although B cells really do not present the antigen for the presentation's sake or for the training of the immune system sake, B cells actually present the antigen to say, look, I have an antigen, now activate me so I can work on creating antibodies. So they're not actually presenters to others. So anyways, so dendritic cell and the macrophages, they have done the presentation. We know that with this, then what happens is some cute little naive T cell would come in and attach. So this is a T helper cell. One clarification I want to do here. Many times people ask me that, hey, would the helper T cell route be taken, or cytotoxic T cell route be taken, or some other flavor of this statement, that would this be helper or something else? So please remember, whenever the adaptive arm starts, it always starts with helper. It does not matter which pathway is taken. So if, let's say, T helper 2 conversion occurs, so this T helper naive cell becomes T helper 2, and then it causes a B cell to become a plasma cell. And I hope you're okay. I'm kind of going through this fast because I think for the whole year we talked about these things. And then if it becomes the naive T helper cell, if it becomes T helper 1, so I have headache with this vaccine shot as well. And so this is let's say, a T cytotoxic T cell, correct? So this is cytotoxic, CTL we call them, or CD8 positive. So now please, at least let's make sure that we agree that helper T cells will perform their duty regardless of where the immune system goes. That is, it goes humoral arm or cytotoxic arm. Either way, helper T cells are involved. So helper T cells are not an option. For example, plasma cell can be an option, that we don't go B cell, we go T cell, cytotoxic T cell. Or it is possible that we don't go cytotoxic T cell and we go plasma cell. That could be an option. But helper, we cannot avoid helper cells. So I wanted to make sure that we are clear on that one part. Now, these plasma cells are going to make antibodies. And we know this, that these plasma cells in the beginning, so here is the important part to keep an eye on. Plasma cell in the beginning will make IgM and they make IgD. They always make IgD. They use IgD as a receptor on their surface. So they always are making IgD. But they would make IgM first. Now this cell, again, this is not new. We've talked about it. Some copies of this cell will be stashed away, will be put in a filing cabinet as the memory cells. But most of the time, the memory cells are of the IgG type. I want to ask if you are able to hear correctly. My computer seems to be slow. Okay, so are you able to hear me? So when I'm drawing, my computer is lagging, so I do not know if it is at least presenting correctly or not. I'm going to continue. Okay, so good. So memory cells will be formed, and then the B cell here, the plasma cell here, this plasma cell, as we keep putting more cytokines on it and say we need more, we need more antibodies, it would say, you know what, fine, I'm going to class switch myself, and it would class switch. And the way to remember the class switch, we have done this before as well. Imagine there is a doctor, her name is MD Gia. So the IgM, then D, which is usually together, then G, then E, then A. That is the class switching route. So this B cell is going to class switch. And what does class switching mean? Class switching means that in the DNA of this B cell, so let's say this is the B cell A. In the DNA of the B cell, all of our cells' DNA, but this DNA is open in the B cell. It has a gene pattern that helps, the gene pattern that helps make IgM protein. After that gene pattern is another pattern that helps make IgG protein. And when a cell is, and imagine if at the end of every gene, there is a wall. So when you are expressing this gene, expressing the gene means you make a messenger RNA from that gene, and then you send that messenger RNA outside of the nucleus. It goes out and connects with the ribosome. We have done these discussions as well in the past. It connects with this cute little ribosome. Ribosome goes over that messenger RNA and makes the protein that the messenger RNA is talking about or has written about. So here, when we are copying the gene here, there is a full stop here, which tells the copying machine, the enzyme that is making the messenger RNA, it tells that enzyme to stop here. Don't go any further. Because of that, the cell would only make IgM. But now let's say the cell wants to do a class switching. What it would do is this. It would literally, the enzymes would come in and they would remove, they would splice off, they would cut off this gene that makes IgM, including this wall. And so now when so this is gone and this piece is connected here. So now when the enzyme will come in to create an antibody, it would actually start from here. And now this would make a messenger RNA that is to make IgG. So when that messenger RNA would go to the ribosome in return will make IgG. This process is called class switching. A cell that has class switched, a cell that has moved from one class to the next cannot go back to the previous class. For example, a cell that is making IgG cannot go back and say, you know what, I want to start making IgMs now, because it has already spliced off the DNA and thrown it out. This is like if you have a recipe book and you have, let's say you're making chicken, some chicken recipe A, and then there is a chicken recipe B, and then you make the chicken with the recipe A, then you tear off the page of that recipe and throw it in the trash, and it is gone. And then you say, you know what, I really want to make recipe A. You cannot make it anymore because that page is gone. So this is what is called a class switching. So from here, we class switch. So if you see back here, so we class switch to another B cell, which would now start making IgG. We will make memory cells of this as well, so that in the future, when we need, it can rapidly start making IgGs. So for example, in my case, I got my shot this morning, second shot, and people are saying, hey, tomorrow, the nurse who gave me the shot as well, they said, tomorrow you would have more symptoms compared to today. Why? Because it takes about 24 to 48 hours for these memory cells that are stashed away in various lymph nodes to wake up, realize that there is a antigen, and start making antibodies. So they would start making antibodies tomorrow, and the inflammatory process would become active tomorrow, and that is when the symptoms may start appearing. So now the IgG is formed. This IgG is what we are talking about today. This IgG is a more durable response, usually an immediate response. Imagine a knee-jerk reflex from the immune system is to make IgM. And then a more specific, a more durable, a more targeted, a more longer lasting response comes in, and that is in the form of IgG. Now, immune system, before I go there, we also know that IgG can cross placenta and go to the baby as well, or the developing baby, to provide the baby immunity. And baby is actually born, when it is born the first time, it's always born the first time, the baby is born with the antibodies given from the mother. And baby lives off of that protection for some time. So imagine that the baby is given IVIg at the time of birth or during the development in the mother. So the initial days of baby, or even months and weeks, the baby's protection is actually by mother's Ig. That is exactly the same phenomena that would happen when we give IVIg, and I'll explain that. So once the IVIg, I keep calling it IVIg, IgG is formed, now that would start taking care of the infection or antigen. Then some of these cells would further class switch, copies of the cells would class switch, and they would start making IgE. Then some more would class switch and they would make IgA. IgA is necessary to protect the mucosal surfaces. So the end result is that as an antigen, for example, nowadays SARS-CoV-2, when that enters our body, we kind of create defenses inside the blood, inside the interstitial tissues or the tissues of our body, and on the surfaces. And we try to kind of protect us from all sides. This is the basic idea of the importance of IgG. So now let's talk about what is the importance of IVIg. And here is an important part, which is, I think the key thing to understand is the following that I'm going to discuss now, and then the rest of the things. The key thing to understand this is that to make IVIg, what we do is, let's say we have a big container. Many thousands of liters of fluid can be in it. In this container, we put people's blood, so the donated blood, and this blood that is donated has a lots of criterias. For example, if you are going to donate blood that would be used for making IVIg, then you would have to donate twice in 6 months. And if you do not donate twice in 6 months, then they would throw the previous plasma, because they want to test the blood twice to make sure that it is clean and it is safe and it doesn't have viruses and it doesn't have any other things that they do not want in the IVIg, which is given to people. Similarly, there are once the blood is collected, it is kept for 60 days, and there are so many important things there. For example, it is incubated there. We may be able to see any pathogens that have incubation time. Then they are going to see if there is anything else that happens. So there is a lot of processes, but I'm going to simplify this for us. So we take all those blood donations, usually anywhere from 1,000 to 60,000 people's blood or those blood bags are added to this pool. Usual practice is 2,000 to 10,000, and I think a common is to use 10,000 people's blood. Now, here is the important thing. Why is it? Whenever I'm reading various articles about IVIg, most of them are more directed towards the patient to say, this is the use of IVIg, you would get it this way, you'll go to the infusion center, they would do this. And they don't talk about why IVIg is actually important. IVIg is important for the following reason. Imagine we have a society where we have taken one, and imagine these are every one person here represents thousands of people. So imagine we have taken people from a society. Of course, all of them have been exposed to various pathogens. So I am sitting here in Cupertino, California. My environment here and the exposure to various pathogens is different. My health is different. My exposures are different. My interactions are different. My hygiene habits are different. So I may have some IgGs. So let's say I may have IgGs. So for example, with the SARS-CoV-2 shots now, I have IgGs against SARS-CoV-2. Then there is someone else who has IgG against some pneumonia pathogen. Then there is someone who has IgG against, let's say, Staphylococcus or Streptococcus or some other antigen, and so on. So what we do is we give IVIg to patients because they are unable to make these antibodies. So imagine maybe I have some immune system deficiency. And remember, it is not only used in the deficiency of immune system, it is used in rheumatology as well, it is used in neurology as well, and so on. So here we have various people, 10,000 people. They have various kinds of exposures and diseases, and against which they have made their own IgGs. And what we do is we take their blood and we put that in here. So imagine now there are 10,000 possible variations and different kinds of IgGs. Normally what happens is, check this out, 1 gram of IVIg, that bottle that has IVIg, it's a big bag, I shouldn't call it a bottle, but anyways, let's call it a bottle. 1 gram of IVIg has 4 multiplied by 10 raised to power 8 IVIg molecules or proteins. And it can protect against about 10 raised to power 7 kind of antigens. So about 10 million types of antigens or antigenic patterns or epitopes can be covered by this, which covers a lot of type of bacteria and virus. So at the end of the day, this is the key factor to understand for the IVIg. Why do we make IVIg? Because we are collecting various, imagine convalescent plasma. So instead of just taking that from one person, imagine you take 10,000 people and take antibodies from all of them. They would have antibodies against a variety of diseases. And then you put them all together. And then from this pool, you take out a bottle of IVIg. Imagine now these IVIGs or these IGs, immunoglobulins, that would be taken out. Imagine how many diverse kinds they would be, and imagine how much they can cover. So this is the basic idea, the benefit of it. Now, generally, what happens for the production of the IVIg or intravenous immunoglobulin? Look, number one, to collect blood from, let's say, 10,000 people, to then process that, to then make those infusions, it is logistically a very rigorous elaborate process. We have to make sure that it is safe. We have to make sure that there is no viruses. We have to make sure there are no bacteria and so on. So it is a very expensive process. It is a very expensive product. Now, the general steps for the processing is the blood is collected. Then from the blood, we do centrifugation of the blood. And what we do is we separate out plasma from the blood and the cells or the RBCs. So the plasma that is separated, that is taken up, frozen, left in the fridge for 60 days. Then it is broken up into pieces. Then what they do is they have this plasma go through so many steps to remove. What are they trying to do from here? Number one, they're trying to remove all other proteins and particles in the plasma. So that is one. So they filter out and they just want the IgGs in it. But generally, they cannot just end up with the IgGs. What happens is after the first filtrations, they still end up with IgG. Possibly there could be viruses. Possibly there could be IgA, IgE, and so on. Then what they do is they once again precipitate these things and they try to remove IgA and IgE. They just want IgG. So normally in an IVIg infusion, about 1 to 3%, and maybe this is refined even more, but 1 to 3% may be IgA and IgE. So they're not a lot. So now the IgG is there, and maybe there are viruses or other things. So then they change the pH, and then they further do photo treatments of this, eventually to the point that viruses are removed as well or inactivated. They put detergents on it. For example, if somebody's blood has SARS-CoV-2, the detergent, so that is an enveloped virus, correct? So the detergents usually kill or inactivate the enveloped viruses by breaking their envelopes. So they do so many things. They change pH, they put detergents. So once they put the detergent, now their second problem is to remove the detergent. So detergent went in, it has done its job of killing the enveloped viruses. Now they want to remove the detergent. So how do they wash IgGs, which are all contaminated? So these are the processes that are done. And there are rigorous steps and there are rules and regulations around this, because we cannot be giving an antigen in virus or, for example, HIV through IVIg. So these are pretty safe. Okay, so that is the process of making IVIg. Take 10,000 people, take their blood, from the blood get their IgGs out that they have made. Now, of course, this IgG that has been gotten out, this need to be preserved, correct? So there are going to be now the problems. We have to preserve this IgG. So there are preservatives that are added. There are fluids that are added. There is sodium that is added. In the old times, in the preservatives, there used to be sucrose-like preservatives. But the problem is they cannot be given to diabetics or they can cause issues. So nowadays, they use amino acids, for example. Similarly, for the other preservatives, they also have the various possibilities, various formulations to target them to various kind of patients. Somebody is diabetic. Somebody may have hypertension. They cannot take too much sodium. Somebody may not be able to take a larger infusion because their cardiac system is not able to handle that, or their renal system is not able to handle it. So there are so many ways that formulations are done to target them to specific people that you cannot say that every single IVIg is going to be the same. They all are different according to what is the formula there. So preservatives are put in there, and then these IVIgs are preserved. Finally, what happens is that they are given as infusions. So patient goes to the infusion center, they create an infusion, and that infusion is given. Nowadays, there are subcutaneous as well. So subcutaneous, that means these are given under the skin, in the skin layer. Now the question, I think this becomes clear, that what is the function, what is the benefit of IVIg? The benefit of IVIg is that somebody who cannot manufacture IgGs in the sufficient quantity or with a sufficiently correct structure, that they can attack the antigen, such folks would benefit from IVIg. Similarly, if there are people who have autoimmune diseases, there may be IVIGs given that can go and attack those antibodies that are attacking their nervous system or their joints or their other tissues. So there are plenty of uses. It's just that because it is an expensive product, it is not used that commonly. Plus, this is a foreign antigen. We're still taking an antibody from a different person and adding it to another person. So that is done. Now the question, let's see if there is any question. Yeah, so they need to be stored. Somebody said here, so they're talking about some of the temperature, but they need to be stored. The IVIGs are usually stored at minus 80 degrees centigrade. Okay. So to give you some examples, for example, when somebody says that, hey, I have long haul and I took IVIg and it helped. What does this tell us? Number one, this person was not able to make an Ig, immunoglobulin G, that was needed by themselves. So that is one. And I wanted to talk about one more thing, and that is the longevity of the IVIg. When you give the IVIg, how long would it stay? Normally, IVIg can stay for 21 days or 20 days. This is why usually it is given monthly, but nowadays with the subcute, there are even weekly doses as well. So going back here, how does it help? So let's say somebody is long hauler. They took IVIg and they're feeling better. Why are they feeling better? There are two possibilities. One possibility is that, let's say there is that zombie virus remnant running around. So why is this zombie? It does not have the RNA in it. So I wanted to do that paper with you as well, where they show that it is possible that the viruses are present, the SARS-CoV-2 is present, but without its RNA. And that SARS-CoV-2, I call them zombie. They don't call it zombie. I call them zombie just for clarity. Those zombie are virus particles, could be just present in various tissues and cells, and that is what can cause long haul symptoms. So imagine if I take this RNA out, can some KoolBeen think about how can that actually be possible? How can we end up with a virus which does not have an RNA but is there? So while you're thinking, so here is a zombie RNA, zombie SARS-CoV-2. And let's say it is just sitting in there, it is getting, they say it does not replicate, of course, because there is no RNA in it. But it is possible that it is loaded in various cells, or it is copying with the cells as the cell divides. For example, a macrophage or a monocyte or a stem cell, when that is dividing, or we call them proliferating, then it is possible that the components of the cell, which include this kind of a virus, I shouldn't call it virus zombie, could become copied as well. But it does not have the capability of replicating like actual SARS-CoV-2. So imagine this virus is present and we want to remove it, and our body has not removed it. So if you give IVIg, it is possible that that IVIg is going to bind with this one and clear it out, like mop it out. So that is one possibility. The second possibility is that, let's say the person has become long haul with some sort of antibodies that are directed against their tissues. And maybe that was because of the inflammatory cytokines that are present, and those cytokines continue to poke the B cells, and the B cells just continue to make bad immunoglobulins. And now you give the IVIg from outside, and it is possible that that would go and attack this immunoglobulin and clear it out. So there are multiple possibilities. Maybe there is actually a persistent virus, although saying that there is a persistent virus, there is no study yet that shows an active persistent virus in a long hauler. But if we assume it for a second, just conjecture that there is an active persistent virus, then giving IVIg would also help clear that virus out. So this is the importance of IVIg. Please remember, IVIg, or immunoglobulin itself, one single immunoglobulin, is not able to attack every kind of antigen. It is just that we collected it from 10,000 or 20,000 or 30,000 or 60,000 people. We get a huge variety of immunoglobulins that can attack a huge number of antigens. So that is the discussion for today. I'm going to see if there are any IVIG related questions, and then we'll do chit chat afterwards. So Arun, you know that IVIG was done. You were one of the persons who wanted to know more about it. So Arun says, answer drug-induced viral assembly problem. So no RNA in viral particle. Absolutely correct. Absolutely correct. So it may be drug-induced or not. It may be our immune system responding, or it may just be, for example, that there can be so many reasons. For example, let's say we have a cell here. This cell is making viruses, and then the raw material for virus finished, and now we have some bodies of the virus formed, but there is no raw material for the brain, and so the virus is going to end up without much RNA. Similarly, if the RDRP is not working correctly, which is responsible, for example, replication of the RNA, then that's not going to work as well. So there can be many reasons that while the assembly of the virus is happening, the RNA is damaged, or it is not correct, or it is not even present. And so this virus could not get its brain, and it is just sitting there zombie-like state. So very good, Arun, you should actually get an award for this. So I don't see any questions. So why not we do this then? Let us hang up for now, and I would come back in the 5 minutes, and we'll do a chit chat. So there is one question I just saw. So Delilah Davis says, can IVIG repair the immune system of a long hauler after several treatments? So ideally, IVIG does not do anything to the immune system itself. Imagine this is like monoclonal antibodies, but instead of being monoclonal, these are polyclonal, and these are just going to sit in the person's body for 10, 21 days, help him with the antigens themselves, and then these will be degraded, and then the person would need them again. So the IVIG itself, it can help immune system in one way, and that is if there are bad antibodies being made, autoimmune antibodies being made, IVIG antibodies can go and attack them as well and kind of clear them out. And that is how it can help reduce the autoimmune disease progress, but not always. It's not necessary that every IVIG has the antibodies against some autoantibodies that we have formed. But it does not repair the immune system itself. It kind of helps take care of the antigen or antibody that is become rogue and is antigenic. Good. So I'm going to stop here. William, thank you very much for the super chat, and yes, thank you very much for the second shot as well. Mr. J says, can you mention again how losing smell and taste might indicate nerve damage or olfactory system? So yes, we can. Remember last time when we talked about the losing taste and smell, I didn't say that there is a nerve damage. What happens is the study that I showed, and there may be more studies showing different mechanisms, but the study that I had showed, that study showed this thing. So let's say this is the olfactory bulb. And so olfactory bulb is in the roof of our nose, and it is going to be doing smell thingy, right? And then around this olfactory bulbs, little nerves that are coming down or branches, there are epithelial cells. And these epithelial cells are making the tissue, this tissue. What they found was, the researchers, they found that inside these cells, epithelial cells, there was infection, SARS-CoV-2, which caused local inflammation, which in turn caused these nerves to be squeezed, and there were inflammatory cytokines over there, and that caused the nerves to be not working correctly or working incorrectly, and that is what caused either those smells that were not really present, phantom smells we call them, or smells just no smell at all. So Mr. J, that is what was the Lonnie says, how do doctors use this for diagnosis, like in autoimmunity? Lonnie, use what? Why not let's do this. Go into the next discussion and we would talk about it over there, just so that this video can then be, contained to the IVIG. Cool. So please like, subscribe, and share. And if you would like to support this work as well, there are 3 links. If you don't want to use the PayPal, there is a link to buy me a coffee. Otherwise, there are 2 more links. One is to become a patron. And I am so happy that now I think we are at 1,100 or 1,200 per month. So thank you very much to all the patrons. And then there is also another link where you can just support this work in general. With this, I would see you in 5 minutes. Thank you.